Glycolysis. Krebs cycle. Long ago, the body's energy systems worked together in harmony. Then, everything changed when metabolic dysfunction attacked. Only the avatar, master of all three stages of cellular respiration, could generate enough ATP to restore balance. But when the mitochondria needed the most, they vanished. 100 years passed, and I discovered the new avatar, a molecule named Megaang. And although she is skilled, she has a lot to learn before she's ready to master cellular respiration. But I believe Megaang can save metabolism.
Hello there, my name is Megaang, and I'm the last Metabender and the Avatar. And today, I'm going to talk about something very important, cellular respiration. It is the force that keeps harmony and powers everything we do. Without it, nothing would really work. Because, as you can see, how I'm supposed to bring balance to the world, cellular respiration is a process by which the cell breaks down one glucose molecule in the presence of oxygen to produce ATP, which serves as the energy currency for cellular functions. Now, to get it all back in balance, I need to master the 3 steps of cellular respiration. First, I will need to visit the nation of glycolysis where glucose is converted to pyruvate in the cytoplasm. Next, I will need to visit the nation of the Krebs cycle or the citric acid cycle. This nation is mitochondrially based and produces high energy molecules such as NADH and FADH2. Finally, I will need to visit the nation of electron transport chain deep inside the mitochondria where the magic happens. Here, the high energy molecules are employed in order to make the majority of ATP through oxidative phosphorylation. I know it sounds like a long journey. I mean, it really is, but every nation has its important role. Without them, the energy balance would not be complete. So, here's my plan. My friends Afghani, Jad, and Justin will first introduce to us the glycolysis, and moving on to the next nation, together with David and Chris, who will shed some light on the Krebs cycle and will highlight the production of electron carriers and released carbon dioxide. Lastly, Randler and Shaina will review the electron transport chain.
The initial stage of cellular respiration is glycolysis, which takes place in the cytoplasm. As this process does not require oxygen, it is classified as anaerobic. Glycolysis produces 2 pyruvate molecules, 2 ATP molecules, and 2 NADH molecules. The first phase of glycolysis is called the preparatory or investment phase. The first step is called phosphorylation. This step, an enzyme, hexokinase, add a phosphate group to carbon 6 of glucose, turning it into glucose 6-phosphate. This process uses one ATP molecule to provide the phosphate group. Second is the isomerization, where glucose 6-phosphate is converted into fructose 6-phosphate in a reaction catalyzed by the enzyme phosphoglucoisomerase. Moving forward to the third step, which is the second phosphorylation. Anaerobic phosphorylation occurs this time on the hydroxyl group of carbon 1, producing fructose 1,6-biphosphate. This reaction is catalyzed by the enzyme phosphofructokinase 1 and uses one ATP molecule to supply the phosphate group.
Next is the fourth step called cleavage. In this step, the molecule is ready to be cleaved into smaller ones. The enzyme fructose biphosphate allyase splits fructose 1,6-biphosphate into 2 molecules, glyceraldehyde 3-phosphate or G3P and dihydroxyacetone phosphate or DHAP. Moving on to the last step in the first phase, which is the conversion of DHAP to GADP. Dihydroxyacetone phosphate or DHAP is converted into another molecule of glyceraldehyde 3-phosphate or G3P by the enzyme phosphate isomerase. This results in a total of 2 G3P molecules. Now, moving forward, the next 5 steps is the second phase of glycolysis, also known as the payoff phase. This phase is where the energy will be produced. Step 6 is called oxidation. In this step, both glyceraldehyde phosphate are oxidized to 1,3-biphosphoglycerate by dehydrogenase. This step produces 1 NADH for each oxidized glyceraldehyde 3-phosphate for a total of 2 NADH. These NADH are later used to produce more ATP for the cell. Moving on to the step 7, which is known as dephosphorylation. A kinase transfers a phosphate 1,3-biphosphoglycerate to ADP to form ATP and 3-phosphoglycerate. This step is reversible even though ATP is formed.
The next step, phosphate transfer, involves a mutase reaction. It moves the phosphate in the third carbon of 3-phosphoglycerate to the second carbon position to form 2-phosphoglycerate. In the dehydration, which is the ninth step of glycolysis, a lyase reaction moves water from 2-phosphoglycerate to form phosphoenol pyruvate. Finally, in the last step of glycolysis, which is the second dephosphorylation, a kinase reaction removes the phosphate group from phosphoenol pyruvate and donates it to ADP to form ATP and pyruvate. With that, 2 pyruvates, 2 ATPs, and 2 NADPH are formed, where pyruvates and NADH could be used in aerobic respiration to produce more energy for the cell.
The Krebs cycle, also known as the citric acid cycle, serves as the second stage of cellular respiration. This cycle is a chain of chemical reactions that release energy in the form of ATP, 8 enzymes that completely oxidate acetate into acetyl-CoA or acetyl coenzyme A.
Unlike glycolysis, the Krebs cycle is an aerobic process, meaning it requires the presence of oxygen, and it occurs in the mitochondrial matrix. The Krebs cycle, however, utilizes the 2 molecules of pyruvate produced during glycolysis.
Before the cycle itself, pyruvate produced from glycolysis enters the mitochondrial matrix. Here, in conjunction with the oxidation of NAD+, it breaks down through decarboxylation by the pyruvate dehydrogenase complex, attaching it to coenzyme A, forming acetyl-CoA. From here, acetyl-CoA will undergo complete oxidation through the Krebs cycle in a series of 8 steps.
The first step of the Krebs cycle is a transfer of acetyl group. With the help of an enzyme called citrate synthase, a 2-carbon acetyl group from acetyl-CoA transfers to the 4-carbon acceptor compound oxaloacetate, forming a 6-carbon citrate compound.
Next is isomerization. With the help of the enzyme aconitase, a water molecule is removed from citrate, resulting to cis-aconitate. Add another water molecule being added again to form isocitrate.
The third step is the oxidation and decarboxylation. Isocitrase is oxidized by NAD+, forming oxalosuccinate and producing NADH. Oxalosuccinate is then decarboxylated, forming alpha-ketoglutarate and producing CO2. All these are possible in the presence of the enzyme isocitrate dehydrogenase.
Fourth is rejoining with CoA. By the enzyme ketoglutarate dehydrogenase, alpha-ketoglutarate is oxidized by NAD+, producing NADH, and joins with coenzyme A to form succinyl CoA, with CO2 as a byproduct.
Now, moving on to the fifth step of the cycle, conversion to succinate. In this step, succinyl-CoA undergoes a reaction catalyzed by the enzyme succinyl-CoA synthetase. During this process, coenzyme A is released and a molecule of GTP is synthesized, which is quickly converted into ATP. This step marks the only direct generation of ATP or JTP in the Krebs cycle.
The sixth step is oxidation to fumarate. The enzyme succinate dehydrogenase oxidizes succinate into fumarate. As this happens, 2 hydrogen atoms are transferred to the electron carrier FAD, forming FADH2, which will later contribute to ATP production during the electron transport chain.
Next is the seventh step, hydration to malate. Fumarate undergoes hydration, meaning a water molecule is added to it, forming malate. This reaction is facilitated by the enzyme fumarase, setting the stage for the final step.
And finally, we reach the eighth and last step, regeneration of oxaloacetate. Malate is oxidized by NAD+ through the action of the enzyme malate dehydrogenase, producing NADH and regenerating oxaloacetate. This oxaloacetate is then ready to combine with the new acetyl group, restarting the cycle.
In summary, the Krebs cycle not only generates ATP but also produces high-energy electron carriers, NADH and FADH2, which are essential for the electron transport chain. It's a remarkable and efficient process that powers life at a cellular level.
Thank you for joining us in exploring the Krebs cycle. Remember, understanding these biological processes gives us a deeper appreciation of how life sustains itself.
Hey curious, and good luck to your next journey.
Welcome to the last but most beneficial stage of cellular respiration, the electron transfer chain, ETC for short.
Actually, it's called oxidative phosphorylation because the process includes two parts, the electron transport chain and chemiosmosis. It generally takes place in the mitochondria, specifically in the inner mitochondrial membrane. Keep that in mind next time.
But you forgot to mention that the ETC is an aerobic process, which means it needs oxygen. And did you know that its main goal is to produce ATP, which we all know is the energy currency of the cell, by utilizing the high energy electrons carried by NADH and FADH2. From the Krebs cycle, of course. Now, let us begin the process. With NADH giving up an electron to complex 1, then it gets super excited that it pumps protons to the other side. That will be useful for later.
Can you at least give me some context? The process contains a series of protein complexes embedded in the inner mitochondrial membrane, known as complexes 1, 2, 3, and 4, which pump protons when it is charged by electrons coming from NADH. At some point, there are also other proteins such as COQ and cytochrome C to help with the electron travel. Imagine it like an MTR with many stops.
Okay, so from complex 1 to COQ, then another electron is used coming from complex 2 to transfer the original electron to complex 3, then passes it to cytochrome C, and lastly to complex 4. All while pumping protons outside the membrane. I guess that is why they call it a chain. All parts are very connected. Once the gradient is established, the protons flow back into the matrix through a protein called ATP synthase. Take it away, partner.
As my acquaintance have said, we shall proceed to part 2 of oxidative phosphorylation, known as chemiosmosis. It powers ATP synthase to combine ADP and inorganic phosphate into ATP. At the same time, oxygen plays a crucial role as the final electron acceptor. It combines with low-energy electrons and protons to form water as a byproduct, ensuring the chain continues running smoothly. To sum it up, the electron transport chain generates around 34 ATP molecules for each glucose molecule, making it the most efficient part of cellular respiration.
So if we combine the net ATP produced by the whole cycle of cellular respiration, we get about 36 to 38 ATP produced. This really brings mitochondria, powerhouse of the cell, to a new level. And I guess that marks the end of this whole process and the end of the video. So make sure to like and subscribe.
This is not going on YouTube.
Now, I have mastered how all of these stages come together to make the world's energy systems run smoothly. Each one of them is important, and together they create balance, and that is why I'm confident we can keep the world's metabolism in harmony.